table we were working with. And in this table, there are a lot of information. The first thing you need to do, based on prior information about how you set up the cross, is identifying the parental class. And usually, the parental class is the one with the largest number of progeny. And so you identify the parental class, and then you identify the double crossover class. Now remember, crossing over occurs at random, and the frequency of two crossovers in the same spot is lower than the frequency of one crossover. And so the double crossover class is usually the class with the smallest number of progeny. So you take this genotype, or the phenotype, sorry, and this, and you basically use this information to figure out the order of the genes. That's the first thing you set out, basically, is to figure out the order of the genes. You identify the parents, you identify the rarest class, which represents the double crossover class, and now based on this information, you should be able to figure out the order of the three genes that you are studying. And the reason for that is that each of these parents, the parental class, are reciprocal. So they are either a plus or a minus, and they have a reciprocal relationship. And when you place three genes in three particular orders, and you put two crossovers between them, the double crossover class will arise. But that double crossover class is unique, depending on the order of the three genes. So, imagine, skip all this. Here are three different possible orders of the genes. You can have vermilion, cross-veinless, and cut. Or you can have vermilion, cut, and cross-veinless. Or you can have cut, vermilion, and cross-veinless. Now each parent for the three genes were different. The first parent was vermilion minus, cross-veinless plus, and cut plus. The other parent was the reciprocal, which was vermilion plus, cross-veinless minus, and cut minus. That order, the phenotypes of those parents remain. It's just the order of the genes that's changing. In this order, the second order, this parent, parent 1, is still vermilion minus right here, cut plus like here, and cross-veinless plus like here. And the second parent is still V plus like this V plus, CT like this CT, and CB like this CB. So you maintain their phenotypes, but you rearrange the order of the three genes. Now once you've done these three different orders, you insert what might be double crossovers. One crossover between vermilion and this gene, the next crossover between this gene and this gene. You put two crossovers, and then you resolve the crossovers. You see what happens when you have a double crossover. So if you have a double crossover, one crossover here and one crossover here, what would the progeny look like? This is the parent, and this parent is going to go through two crossovers. So if you have two crossovers, what are the progeny classes going to look like? So you just basically walk along here. Here's little v, v minus, crossover, cv minus, crossover back up, cv plus. And so you write down what we expect. So these are the predicted double crossovers based on the order of the genes. If the order of the genes is in this fashion, then the double crossover class will look like V, CV, and CT plus. And the reciprocal will be V plus, CV plus, and CT. If, on the other hand, the order of the three genes is here, then the double crossover class will look completely different from this double crossover class. Each of the double crossover classes that you predict are going to be unique. They will be distinct. So in this instance, you get a V followed by a CT followed by a CV plus, V plus, CT plus, CT. And in this third instance, you get double crossover class that is CT plus, V plus, CV plus over CT, V, and CT. Notice these 6 double crossover classes. None of them are identical to one another. class is unique, VCVCP plus. I don't see any VCVCP plus anywhere else, do you? No. That's the key. Because each of these predictions are unique, you take these predictions and you ask what is the observed double crossover class. And the observed double crossover class is right here, V, CV plus and CT. So where is V, CV plus and CT? Here is V, CV plus CT. So because that's the observed class, this must be the order of the genes, because that's the only way the observed class would appear, is when the genes are in this order. And so you identify the order of the genes even before you do any calculations. Once you've identified the order of the genes, then it's just a matter of adding the numbers and you get the distances. So it's a very quick process. Instead of doing 3 different 2-point crosses by separating out the third class and calculating the differences, you can do the entire calculation once you know what the order is. So here is the original genes. I've just written them as B, C, D, C, etc. You basically now know what the order of the 3 genes are. The order of the 3 genes are B followed by CT followed by CV. So this class here, this phenotype class, V, CV plus, CV plus, you just can rewrite it as V, CT plus, CV plus. Because you're just rewriting it, you're not changing anything. So V, CV plus, CT plus is basically B followed by CT plus followed by CV plus, and that's 377 progeny. And then the reciprocal B plus C plus CV. This class here, B, comma, CV, comma, CT plus, is actually along the chromosome B dot 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 CT plus dot 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 CV. This is now the order of the 3 genes on the chromosome. And so on for all of them. Okay. So now that you've identified the order of the genes, you know B is followed by CT followed by CV, you label them. Here is crossover 1, and here is crossover 2. If you had a single crossover, just one, let's imagine you have a crossover only here, you would get a class that would look B, CT, CV. And that's this class here, V, CT, CV. So this 75 units comes as a number of crossovers that are taking place here. This 43 units comes due to a single crossover that happens here. And so then you just add the formulas up. So there's your parental class. This is a single crossover here. This class here. This is the single crossover that takes place here. Right there. And then this is the double crossover. So what's the distance between the genes? Well, it's always recombinance over progeny. So in this instance, for this class here, it's SCO1 plus DCO over the total. You add the double crossovers to the single crossovers because there was a crossover here in this class. And it was right there. And so you add that to the SCO1. Similarly for SCO2, the distance between CD and CD, that's SCO2. It's actually the distance is SCO2 plus DCO over the total. So you add the double crossovers to both sides, to the single crossover 1 and the single crossover 2, basically. And so you calculate that, it becomes 75 plus 75 plus 5 plus 5, which is 160 over 1,000, which is 16 math units, which is exactly the same as what you obtained last time. And the other class is 43 plus 43 plus 5 plus 5, which is 96, which is 926. So you get the same answers with 2 different methods of looking at distances. Okay. So what's going on? Okay, so there's another issue that we have to pay attention to when we're mapping genes, and that's known as interference. So when a cell initiates a crossover at a particular position, is semi-random, happens more or less at random. But once the first cut is made to start a crossover, there is inhibition of crossovers in its vicinity due to changes in the structure of the chromosome. And that's known as interference, recombination interference. So when you have one crossover, there is less likelihood of having another crossover immediately adjacent to it. And that's known as recombination interference. And you can actually calculate the amount of interference between two genes. So here are the map distances. 16 map units between B and CT, and 9.6 map units between CT and CT. So that's the number of recombination events that are taking place here, correct? So the probability, calculated probability of a double crossover is 16 times 9.6. Right? Expected double crossovers is the product of the single crossovers. That's what you expect. So this is of course a frequency, so you can calculate the frequency. 0.16 times 0.096, which is 0.0154. So that's the expected number of double crossovers. Theoretically speaking, if everything is normal, then you should expect to see 15.4 progeny that have gone through a double crossover. Okay. In a total of 1,000 progeny, you should get 15 progeny that would have had a double crossover. What we observed was only 10 double crossovers. This difference is due to interference, recombination interference. And so you can calculate that as a coefficient of coincidence, 1 minus expected over observed. And so that's what you can calculate as the amount of interference taking place in terms of the number of recombination events that should have been observed but were not observed. And the difference is recombination interference. And so you can calculate that 1 minus observed over x. All right, so I can give you another example of a 3-point cross. In this example, we are starting with a parent that looks like this. Different from the first instance. In the original cross, we had 2 pluses and 1 minus, cross to 2 minuses and 1 plus parent. In this instance, there are one parent is all minus, the other parent is all plus for the 3 genes that we are studying. F1 progeny is right here. SC plus EC plus VG, that's that chromosome from this parent, and SC, EC, VG, that's this chromosome from this parent. And you always set up a test cross, so homozygous recessive, and you get progenies. Okay? And you look at the progeny classes. I've written out the progeny classes. Now, if you see a table that looks like this, remember what I told you. Usually when you get a 3-point cross and you get a table, you get 2 individual classes that are very large numbers, and then smaller numbers, and then very small numbers, right? You don't see that here, right? You've got 4 classes that are approximately equal here, and 4 classes that are approximately equal here. That's a sign that only 2 genes are linked, and 1 gene is unlinked. So when you do a 3-point cross and you get numbers that look like this, that should immediately tell you that 2 out of the 3 genes are linked, and the 3rd gene is not linked. And because it's not linked, you're not getting the 2 classes the way you originally received. You're seeing a different pattern. And when you get that pattern, then you have to do what we did initially. You have to eliminate 1 class and analyze just 2 genes at a time and find out which 2 genes are linked and which 2 genes are not, and which third gene is not linked. And you figure that out by looking at the classic method. Okay? So if these genes were on separate chromosomes, they should assort independently. If you were analyzing 3 genes at the same time, all of these numbers would be equal. Okay? So imagine when you are looking at something that looks like this, you basically eliminate the third class, you add the numbers up, like we did before. And you can see immediately that these numbers are not unlinked. These are linked genes. And so you can say SC and EC are linked genes. Correct? Because here is the parental class, 478, and here is the recombinant class, it's only 26. And you can calculate the distance between them, and then it's 5.5 nanometers. And you can say SC and EC are linked. So SC and EC are linked. Then you do the next experiment, SC and EG. Here we go. Classes again. You combine them because you're simplifying. And you see now, you get equal numbers. Here are the 2 recombinant classes here, 257, 249, very similar to the numbers here. So these 2 genes are unlinked. And so you can say, this is the gene that's unlinked. So VG is not linked to SC, but SC is linked to EC. And so if you have 2 chromosomes, SC is on one chromosome, VG is on the other chromosome. And then if you do the third experiment just to confirm it, you find EC and VG are also not linked. So SC and EC are on one chromosome, VG is on a third chromosome. So just by looking at the numbers here, you can tell if 2 out of the 3 genes are linked, all 3 genes are linked, or only none of the 3 genes are linked. What would the numbers look like if none of the 3 genes are linked? So let's pretend we do it across. Here are your 8 classes total. Here are the numbers, just made up numbers, okay? So when you look at this pattern, which pattern indicates that all 3 genes are linked? This one, or this one? Middle one. The middle one looks like a classic 3-point cross. There is one class with very large numbers, one class with double crossovers, and then here are your single crossover clusters. So this pattern of numbers indicates that all 3 genes are linked. This pattern indicates that 2 out of the 3 genes are linked, but the third gene is not linked. And when you get a pattern that looks like this, that means that all 3 genes are unlinked. Okay, all 3 on 3 different chromosomes. So just by looking at this, you can tell immediately which genes are linked. You don't even have to calculate initially. Once you know one is linked and 2 are unlinked, then 2 are linked and 1 is unlinked, then you can go and do the experiment that I showed you to figure out which of the genes is linked. Okay. Any questions about this or about the midterm? You can ask me right now. No?
What was the average?
29 out of 50. But that doesn't mean anything in terms of grades. In this class for genetics, passing grade is 50% or higher. So if you have less than 25 out of 50 on the midterm, currently you are failing. And you need to work harder or slightly different so that you can catch up and make it. The final is going to be very different from the midterm. Question? 49 out of 50. Didn't get to 50, I don't know why. It was one silly multiple choice mistake that was.
To clarify, by passing, do you mean a D or a C?
C. There are some of you who will get D's and that's okay for you because I believe D's are fine for certain classes, for certain tracks. And So yeah, that's different. 50% and higher gets you a C. In terms of it's mostly molecular rather than mathematical. So there's a group of individuals who hate math and love DNA sequences and molecular aspects of genetics. They do really well in the final, but not so well in the midterm. Then there's a group of students that love math but hate the molecular side of it. So they do really well in the midterm, but not too well in the final. And then there's a whole group of very large number that do well in the midterm. So it's kind of there is some crossover taking place. Sorry? 60 will be the final, 40 is the midterm. So even though the midterm right now is 50 points, I will calculate it so that it's equivalent to 40% of the total. The final will be 60% of the total. The size of the final will be almost the same. It will be no more than 10 pages. And you still have, and you'll have twice as much time because you'll have 3 hours. So you will have much more time to find them.
What's the range of an A?
Oh, I have no, I mean, it fluctuates. I tend to curve. So I won't go into that. Do you have any questions about the questions in the midterm? Talk to your TAs, talk to me in office hours or after class, or even now if you want. We can have a 5-minute or a 10-minute on some of the questions. But do talk to them, because there were aspects of the midterm that will show up in the final. Pedigrees will show up in the final. Sequence analysis will show up in the final. So, you know, in the short answers, the sequence analysis questions, that could show up in the final, more detail, more interesting aspects of it. Similarly, pedigrees, but except you'll be using molecular markers instead of phenotypes. So, but you will be doing pedigrees. So if you have difficulty with pedigrees, you need to practice more, ask questions, or talk to TAs, talk to me, whatever, to get better at it. Okay, so we're going to continue with recombination and mapping. So up to this point in the class, I've kind of said that crossing over occurs at random between 2 genes. Well, that was an oversimplification. Now I'm going to take you into the exceptions to that rule. What happens? So crossing over or recombination does not, in practice, happen totally at random. There are factors that affect the crossing over. And so those factors then affect the mapping, or the distance we calculate using this method between genes. Okay? So in most cases, the order of the genes that is revealed by these mapping experiments stands true and stays true. It does not change. So when we sequence the DNA, we determine the order of the genes, and the order of the genes that are determined by recombination mapping more or less match the order of the genes based on sequence. So that's not a problem. The problem is in the distances between. The physical distances between genes do not always correspond to the map units. And the reason for that is what I'm going to talk about now. Okay. Gene order but not gene distance is consistent between the genetic maps and the physical maps. So recombination maps are called genetic maps because they're looking at genetic recombination rates as a function, as a predictor of distance between genes. So, one of the reasons we've already covered in the class, genes that are very far apart will undergo double, triple, and quadruple crossovers. And so they will reduce the number of crossovers, and so they will appear closer than they actually are. And so you have to then take into account the number of crossovers that are taking place. But there are other differences as well. So this one we've already covered because of the double crossovers, masking the recombinant class, you get a reduced number. Here, you also have differences based on species. There is greater recombination in one species compared to another. So in yeast, there is much greater recombination as a function of distance than in humans. And so when you look at map units in yeast or in humans, in human, one map unit is approximately 1 million base pairs. Okay? So if you do an average calculation of distance based on map units and based on the actual sequencing information, one map unit in the human chromosome is approximately 1 million base pairs. The numbers fluctuate, but they're around that number. In yeast, on the other hand, one map unit is only 5,000 base pairs. So there's much greater recombination and crossing over in yeast than in humans, and therefore one map unit corresponds only to 5,000 base pairs. So that's one difference that we observe species-specific. And it's to do with the rate of recombination. Recombination is a function of double-strand breaks made during meiosis. So if you have greater number of breaks, then you will get greater number of recombination. Another difference that you see are sex-specific differences. And some of you have been told there are sex-specific differences because some PA has told you that in Drosophila, in males, there is no recombination. Question?
The question is, I thought recombination happens more farther apart. So why, when these are like closer together, they have more recombination?
What do you mean by closer together?
So the base pair.
Oh, no. So what I'm saying is that there is one recombinant event every 5,000 base pairs compared to in humans where there is one recombinant event every million base pairs. So what is map unit? Percent recombinants. That's what I'm trying to do. So, another difference is sex-specific differences. And some of you have been told that in Drosophila males there are no crossing overs taking place, and that is true. That's an extreme example. So, if you take two genes in Drosophila and you map them, you get a distance that is D12 between two genes, D12S7 and PAH. With map distances, if you measure them via recombination in one instance versus another, is 9% map units in males and 22% map units in females. And that's not observed. That was actually a mistake. It's not in Drosophila. This is this is human genes. So map distances between two human genes, D12S7 and PAH, in males it's 9%, in females it's 22%, or 22 map units. So there's a twofold increase in the distance because there's a twofold increase in the combination frequency in females compared to males in humans. The extreme example is in Drosophila males, where there is no recombination. So you get 0 if you were to measure recombination between two genes in males, because they have no recombination. The way Drosophila males undergo meiosis is kind of interesting and totally different. They use a different aspect of segregating chromosomes, so they don't need to have any crossovers. They stick to the membranes of the nucleus and then they separate out. So here is a map of human chromosome 12. This is what the human chromosome 12 would look like based on distances between genes if you were to map genes in females only. So if you were to take a heterozygous female and cross it to a homozygous recessive, like you would do a test cross, and if you were to do that, this is what the female map would look like between gene here and here, here and here, etc. So you can see this is a female map. It looks much larger because you have greater recombination distances between each gene. This is what the male map would look like for the same chromosome. Because there's fewer recombination events, the distances between the genes are closer together. And so you kind of map them as a smaller chromosome. And so what you usually observe when you draw a map is a sex average map. It's the average map of the female and the male in a sense. So you draw them out like this. And that's one aspect that you observe, is that there is a difference in recombination frequency between the sexes. Another important difference between recombination frequency or crossing over is along the chromosome itself, there are differences. So if you have a chromosome There are regions of a chromosome where you get increased crossing over, and there are regions of a chromosome where you get reduced crossing over. So these are the regions where you get significant crossing over are called hotspots of recombination. And regions where you get very little crossing over are called recombination deserts. So between gene A, B, C, D, and E, you can see there is increased recombination between A and B, so there must have been a crossover here. But there is rarely a crossover taking place between genes B and C.